In this video, we will discuss about this Adeno vector diagram and the transduction pathway and how it will trigger generation of antigen. So we will cover in this video the intracellular pathways. For the extracellular pathways, we have already discussed that thing in our previous video. You can go through that video if you want to understand what are the intercellular pathways. In this video, we will discuss intracellular pathways. So we'll start with this adenovirus vector vaccines. So this is how the adenovirus vectors looks like. So it has this knob shaft which is together known as fiber, and this hexagonal kind of shape, and this penton base. And when it comes to this genomic sequence, we can see that it has this ITR, that is inverted terminal region. So this the ITR is important because it will determine the lifetime of this adenovirus vaccine. And after that, this ITR, it contains this antigen encoding gene, or we can say that the antigen informations that is essential to generate antibodies against the virus for which these adenovirus vaccines are made. It contains the promoter and the antigen encoding gene, and thereafter this poly-A tail to stabilize poly-A tails to stabilize this antigens RNA formations. And in the end also it contains this ITR, that is inverted terminal repeats, not region, inverted terminal repeats. This ITR is essential to, as it will determine the shelf life of this adenovirus vector vaccines. So how it will happen, how it works, and how it transduces within the cells is that this vector come and interact with the receptor that are present on the cell surface, and which are commonly known as the CAR receptors, and this helps the vector to come in contact with this alpha V integrins that helps in this endocytosis of the cells. And thereafter, because of the pH change, the fiber portions got break down within this clathrin coated endocytic vesicles that can be seen here, and after that this hexagon shapes vector is escape into the cytosol. And once it escapes into the cytosols of the cells, it can then direct it towards, it can move towards the nuclear pore which are present on the nucleus, and where the docking at the NPC site, nuclear pore sites, can occur. And once it docked at the nuclear pore sites, it can inject its adeno vector genome. And this adeno vector genome thereafter, since the nucleus contains the necessary enzymes for transcription and translation, so this ad vector genome will then be transcribed and translated within the nucleus, and thereafter the antigen informations will then be move outside. The antigen informations will then be translated after transcription, and then it can be these informations can be provided into the extracellular matrix, where the immune cells will determine those non-self antigens and generate an immune response against those antigens. And this immune response generated by the immune system will be both the innate immune response and the adaptive immune response. In the case of innate immune response, the response is quite general and immediate, or it is the first defense line of the immune system. And the adaptive immune response will be more specific, and it will take time, and those immune response will be specifically targeting the antigens that are been detected by the immune system. And those innate and adaptive immune, how this innate and adaptive immune system works. We discussed that thing in our previous videos. You can refer to those videos to understand this difference between innate immune response and adaptive immune response. So this is a general idea how the adeno vector diagram looks like, and how it helps in the transduction pathways, and what are the roles of this ITR that helps in the stabilization, or helps in the stabilization, at the same time increase in the lifetime of this adeno vector vaccine within the cells or in the nucleus. If you need to understand more about this transduction pathway or adeno vector diagram, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.